here, thank you, sir. Is it recording?
Mm-hmm.
Is it,
Yeah.
Sometimes when it's a long pause, you have to press it twice. But you saw the red, you see the red, is it?
Yeah, it's on.
Okay, great. So we talked about the bones in the neck area. It's called your cervical bones. The bone in your chest area, right here, but in the back. In the back. Those are called thoracic area, and the 5 bones in your back, the lower back, is your lumbar. So, and then sacrum and coccyx, they are also bones, but there is no spinal cord passing through them. But these spinal cord pass through them, and I think it's number 4 or 5. That's where spinal cord stops. Okay. Again, I forgot my anatomy a little bit. And then I gave you the function of atlas and axis and so on and so forth. Enough of information from last material. Okay, bone, we talked about that. The cancellous bone is spongy bone, another name. You remember your compact bone, the bone, like for example, the bones in all of your long bones, most of the bones in your body, except flat bones, they have around it, in parameter, they have the compact bone, and in the middle of it is spongy bone, and inside of the spongy bone, you saw the picture of it, is your bone marrow, which bone marrow is responsible for making white blood cells and red blood cells. Compact bone, I do not have a name for it, but for spongy bone, I have a name called cancellous bone, and osteoblasts, they are bones that are responsible for bone remodeling. They call it bone remodeling. So after age 25, different textbooks say different things, but I say 25. After age 25, you stop growing. You don't grow anymore, okay? But up to age 25, some textbooks say 18, 19, so anyhow. But still, until the day you die, your bone undergo remodeling. And that's what I'm going to talk about next. Your bone undergo remodeling. And the cell that is involved in bone remodeling is osteoblast. The other bone that is osteoclast. The other bone that is responsible for bone remodeling is osteoblast. Easy to memorize this. B, osteoblast, is for building, right here. B for building and class for reabsorption, destroying bone. The 2 hormones, calcitonin and parathyroid hormone. Calcitonin and parathyroid hormone. Calcitonin comes from your thyroid gland right here. It looks like a butterfly. And then in the back of your thyroid gland, there are 4 parathyroid glands. The parathyroid hormone, it makes parathyroid stimulating hormone or parathyroid hormone right here. So what happens with parathyroid hormone? It increases the calcium level in the blood. And what happens? Parathyroid hormone stimulates these cells, osteoclasts, and osteoclasts break down bone. That was a question in the lab. What happens between osteoclasts and osteoblasts, and Ray lost her bone, something like that. You remember that? So what happens? Parathyroid breaks down bone, allows a lot of calcium, and the bone is made up of calcium and phosphorus. A lot of calcium get inside of the blood vessels. Then calcitonin is secreted. When your body senses that there's a lot of calcium in the bone, which is not healthy, so when your body senses that, what happens? Then the cells of your thyroid gland stimulates secretion of calcitonin. And what does calcitonin do? It stimulates the osteoblasts to take up calcium from blood. So it lowers calcium. Calcitonin lowers the calcium level in the blood. And parathyroid increases the calcium level in the blood. How? Parathyroid hormone breaks down bone, and calcitonin builds up bone with help of these 2 cells which are in the bone, and I talked about that. I hope that makes sense. So the level of calcium in your blood constantly goes up and down, up and down, up and down, just like the level of insulin in your blood. It's always up and down, up and down, up and down. I hope I'm making some sense. Okay, so that's what happens, the relationship between osteoblast, the question in your questions at the beginning, osteoblast, osteoclast, calcitonin, and parathyroid hormone. Okay, is there any question about that? If not, let's move to the next thing, which is muscular movement. So I'm all done. I'm finished with everything about muscle. Everything that I supposed to say about, everything that I supposed to say about bone, I said it already. Let's move to muscular movement. So the name of the theory, there are two names for it, sliding filament model, and what happens, atrophy, it means when you lose your muscle cells, they die. Remember, there are two types of tissue in your body that you were born with a number of them. It's called utility, concept of utility in biology. Utility means constant number of cells. Constant number of cells. You were born with a set number of, let's say, 1 million, 2 million, whatever the number is, I don't know, of neuron in your body, and you will die with those number of neuron in your body. You were born with a set number of muscle cells in your body, and then you will die with those set number of muscle cells in your body. Nothing goes wrong in your lifetime. That concept in biology is called utility. Utility means constant number of cells. Okay? True number of cells. So the muscle cells in your skeletal muscle, smooth muscle, cardiac muscles, if they die, they will not grow back. The number of the cells in your brain or spinal cord die, they will not grow back. Okay, so I hope I'm making some sense there. So that's what atrophy means. Atrophy means the muscle cells are dying. That's pretty much what the muscle cells are dying. Then there are two other things. Hypertrophy, it means the size of muscle cells increase. The size of muscle cells increase. Like you go to the gym and you lift weights, and what happens, the size of your muscle cells increase, not the numbers. The numbers do not increase, the size increase. And then you have hyperplasia, it means the numbers increase, and that is not healthy. That could be a cancer. Okay, when the number of the cells, muscle cells increase is called hyperplasia. Hyperplasia is the number of the cells which is not normal, and hypertrophy, it is normal if you work out. If you exercise, you pump iron, then the size of the cell increases. Okay, the other theory, same thing as sliding filament model or attack pole release, they call it. It's about 50 to 100 cycle times per second. And I'll show you, we'll talk about it. Just a few things. Creatine, phosphate, it's a source. You know, athletes nowadays take creatine. In old days, when I was in college, athletes used to shoot steroids, but now later on, I realized, oh my God, what did we do? What did we do? We are not a man anymore. You know, they lose the liver, you know, nothing for it. So that is outlawed, having steroids. But nowadays, athletes are taking creatine. I don't know if you've heard about it or not, in order to save phosphate right here. What happens, phosphate attaches to creatine, and when you exercise, the phosphate separates itself from creatine. The phosphate separates and it attaches to itself to ADT, and ADT becomes ATP, so you can run faster, longer, whatever is the long that you are dealing with. But again, personally, I'm being recorded on YouTube. I do not recommend taking creatine. I don't know. That's my personal opinion. Because right now they don't know the effect of creatine on a person, but maybe 10 years, 20 years, 30 years down the road, they find out, creatine causes, I don't know, make you have 3 eyes instead of 2 eyes. Whatever they come up with. They don't. So I would not take it. I don't know, our soccer player is not here. I don't know. He should listen to the YouTube lecture. But anyway. Red muscles are highly vascularized and have many mitochondria. White muscles, white fibers. Yeah. Okay, you know we talked about this. This is skeletal muscle. There is a striation nucleus on the periphery. This would be back picture of cardiac muscle, and this would be smooth muscle. Nuclei are in the middle of the cell. Nuclei in the middle of the cell in cardiac, and so on and so forth. Okay. So you have a muscle bundle, just like nerve bundle, you guys. You have, if you look at your biceps, you have a bundle of muscle. And I'm not too crazy about these terms, fasciculatus, and so on and so forth. The nucleus are on the periphery, nuclei, I should say, on the periphery of the cell. There are more than one nuclei per cell. So if you dissect, you take up the muscle cell, and then this, the T tubules, I'm not too crazy about it either. It's not even mentioned here. Oh, great. It's not mentioned. These are T tubules. I don't know, it's mentioning a sarcoplasmic reticulum. Yes, T tubules are outside. He's right. This is inside of the cell. I'm wrong. Inside of the cell is sarcoplasmic reticulum. That's right. That's where storage of calcium is. You remember biology 1, you studied smooth ER, smooth endoplasmic reticulum, and rough endoplasmic reticulum. Smooth ER do not have ribosomes. Sarcoplasmic reticulum does not have ribosomes, so it is a type of smooth endoplasmic reticulum from biology 1 note. Of course, nucleus on the periphery. The cell membrane of the muscle cell is called sarcolemma, and sarcolemma are attached to the T tubules, so on and so forth. It's not mentioning it here. I don't worry. I'm not worried about it either. So nucleus, sarcoplasmic reticulum, and then A band. What is the definition of A band? If you look at it, A band, by definition, is where you have actin and myosin. Amir, what is actin? What is myosin? Hold on to your pants. I will go over that in a minute. I don't know what is actin and myosin, Amir. Hang on. Okay? So, but right now, write down in your notes that A band is where there is actin and myosin together. Where they are both actin and myosin. Then I band, that's where only you find actin. Actin, another name for it is called thin. Thin, like a person is thin. This is thin filament. And then myosin, another name for it is called thick filament. T-H-I-C-K. I hope I'm making some sense. So that's another name for actin and myosin. I will talk about that. A sarcomere, definition of a first of all, sarcomere is unit of muscle cell. One unit of muscle cell is called sarcomere. Unit of money in the United States is dollar. Unit of money in Europe is euro, and of course, England always were on their own as pound, whatever it is. The rest of the world, I don't know. For Russia, unit of money is rupee. Is that right? Ruble, whatever it is. I don't know. So anyhow, for India it's rupee. Is that right?
Yeah.
I don't know. Anyhow, you guys are giving me a hard time. But anyhow, from Z line, I will tell you what is Z line in a minute. From Z line to Z line is called sarcomere. Sarcomere is unit of muscle cell. Okay? And then Z line, of course, I band, as I said, which is there is only actin molecule. Are you guys ready? Let's go. So I gave you, this diagram, it will give you, I asked you on the questions at the beginning, it says, what is histology of muscle? This is it. If I ask you, what is histology of muscle? This is it. This is histology of muscle. Skeletal muscle, of course. Skeletal muscle or cardiac muscle, a little bit different, but same thing. Okay? I hope I'm making some sense. Let's go. Here's the skeletal muscle, nuclei on the periphery, lab practical, lab questions, of course. Okay, let's talk about thin and thick filament. What is their anatomy? What is this? So you have in thick filament or myosin, you have a lot of these structures, as you can see, like a golf, what do you call it, club? Is that right? So at the beginning of it, it's called head of the myosin, and this is called tail of myosin, and this is all made up of protein molecule. So in here, in the myosin, you have a lot of these heads and tail. These are all heads, you guys see that? And these are all of the tails. They're all together, like a bundle. Okay? And then the heads are here, the tails are here on both sides. This shows probably half of it, but the other half is right here of the myosin. Okay? And on top of the myosin, these are actin molecules. These two are referred to as actin molecules. Actin molecules, again, they are protein molecules. And then they're like a tennis balls. Imagine these as tennis balls, and wrapped around them are like a rope. These are called tropomyosin, right? These tennis balls are called actin molecules. You guys see, you are following me, am I following myself? Okay, so actin molecules. And then there are ping pong balls scattered throughout this actin molecule. Okay, those ping pong balls are called troponin complex. Still, I'm talking about histology of skeletal muscle. I'm not talking about anything else except histology of, I gave you a rundown of what is actin and myosin is. Okay, and then look, Z-line, you guys remember that? Z-line, I talked about it, from Z-line to Z-line is called what? Sarcomere. Sarco on your prefix suffixes means what? Flesh, meat. Okay, so that's where you got the term from. Sarcomere, from here to here is called one sarcomere. And then, look, the actin molecules, the tennis balls, are attached to Z-lines. Okay, so let's move. So I just gave you histology of everything. Here it is, the polypeptide coil of the myosin molecule. Here it is again, both of them. Here is a troponin. Tropomyosin is the rope, and then actin molecules are the tennis balls, and ping pong balls are troponin complex. Okay, I'm making analogies. You've seen those balloons, they tie them together in the celebrations. I don't know, but add those ping pong balls to it too. Okay, so what happens during muscle relaxation? That's how it is when your muscle is relaxed. This is how it is when your muscle is contracted. Right now, when this is this. That is when I'm relaxed. I gotta play you guys next time. Today I pledge you money. Next time I'll play you guys a relax, don't do it. You've heard of Frankie Goes to Hollywood? Wesley have heard of that. You don't have to play that. It's a nasty song. I don't know. We gotta have fun in here too. We cannot always talk about what was the last time we always talked about poop and urine, I don't know, blood, and the rest of campus is having fun talking about Frankie Goes to Hollywood. That's the name of the band. The name of the band is Frankie Goes to Hollywood. Aren't you ashamed of yourself? I said, no, I'm not ashamed of myself. I'm trying to have fun. Okay, contracted, relaxed. So, let's see what happens. I don't know what happens. I don't like these. I don't like it. Yeah, it doesn't explain anything. Right here, do we have it? Yes, I have it. Great. These are kind of new to me. If you watch, you remember this, guys? Neuromuscular Junction from a century ago, 2 centuries ago, you learned in this class. You remember that? So here's a nerve coming down, and axon terminal release acetylcholine. You remember that? And then when acetylcholine is released to the muscle, that's what we need to talk about today. That's what you need to take home today. What happens between the nerve and muscle? You learned already what happens between nerve and nerve. Now you need to know what is happening between nerve and muscle. So they release acetylcholine, and here's a Schwann cell releasing acetylcholine, and when acetylcholine attaches to the surface of the muscle cell, then what causes acetylcholine when they attach to the skeletal muscle, what causes the, I'm trying to break them, Amir, they're not yours, break. Okay, so what happens right here, you guys see that they release, right, this is the neuron, right? It releases acetylcholine to the muscle, and what happens, the acetylcholine outside of muscle cells is a lot of sodium, just like neurons, there's a lot of sodium here. So all of that, the acetylcholine causes sodium rushes into the skeletal muscle. Just like neuron. Whatever you learn on neuron, you're relearning it again. Okay, so sodium, a lot of sodium gets inside of muscle cell. And then what happens, those sodium, when they get inside of muscle cells, they cause release of calcium from sarcoplasmic reticulum. Calcium is released from sarcoplasmic reticulum. Then they go attach, here they're showing you the release of acetylcholine to the sites, to the receptor sites, right here, the receptors of acetylcholine receptors, and then it causes the sodium channels to open up on the skeletal muscle. Here, the sodium channels open up, and then what happens, I said, I'm not going to talk about tubules, and what happened, a lot of sodium, it gets inside and causes a release of calcium from sarcoplasmic reticulum. Watch. Calcium is released from sarcoplasmic reticulum. Are you guys watching me? Mm-hmm. And then the calcium comes and attaches to troponin complex. He mentioned troponin here, but it's actually troponin, those ping pong balls. They're coming into picture. Calcium attaches to troponin complex. When they attach to troponin complex, what happens to the shape of the actin? The actin molecules changes its shape. Become from this side, they become like this. When it becomes like this, they change, the protein molecules changes a little bit. The head of the myosin attaches to the actin. So what happens, the actin molecules have calcium attached to them, they change, the head of the myosin attaches to them immediately. And then they let it go, they attach to another part. And of course they pull them, attack, pull, release. Sorry, I forgot pull, release. So they attach the head of the myosin, this is the tail of the myosin, attaches to the actin, and then release, attach again, and pull. Release, pull, attach. And that's what is happening right here. But when they are detaching, ATP needs to become ADP. So when the head of the myosin attaches to the actin, you don't need ATP. It's when the head of the myosin detaches from actin, you need ATP. That's why when people die, they become, have you heard of a rigor mortis? Mm-hmm. They become stiff because there is no ATP. When they die, the head of the myosin attaches to the actin, and they don't let it go. And that's why there is no ATP. They are not making ATP. Right now, we are all making ATP in our body. Okay, so we let it go. In them, they cannot let go, and that's what rigor mortis. Okay, so, and this goes on and on and on. Right here is a summary of what is happening. Here is, this is what slide I like, and tells you all of the steps that you need to know about muscle movement. Sliding filament, or attack, pull, release, attack, pull, release, attack. I should get it from here. Attack, pull, release, attack, pull, release, attack, pull, release. So when a muscle is relaxed, it's the sarcomere, Z-line to Z-line. When a muscle is this big, when it's relaxed, the sarcomere is this big. But when it's contracted, a muscle is contracted, its sarcomere is short. Okay, I hope I'm making some sense. And I go back to the original slide and see what happens. Okay, so what happens right here, you guys, let's go over acetylcholine is released, and then the sodium comes inside of the cell. Here is sarcoplasmic reticulum, releases calcium. Calcium attaches to actin molecules. The head of the myosin attaches to the actin, right? And pull them and make them like this. So this is a muscle that is contracted, this is a muscle that is relaxed. So quickly, answer my question. Quickly, what happens to A band when a muscle is contracted? What happens to A band? Does it become shorter? Does it become longer? Does not change at all? What happens? Nothing. Nothing happens to it. It stays same. Remember, by definition, A band is where both actin and myosin are. When you have a muscle contraction, That's where both actin and myosin are, and nothing happens.
Would make it
What?
bigger then?
On a contracted muscle, A band is from where you have both actin and myosin. This is A band, right?
Yeah.
Where you have both actin and myosin, nothing happens to the distance of it. Okay? What happens? Do you remember another set of these are right here? So what happens? Imagine this is shortened. What happens to I band? It shrinks. What happens to, I didn't talk about it, where there is only myosin, it is called H zone. H zone. What do you think happens to H zone? Oh, what happened? Here on the relaxed muscle, you have H zone. Do you have H zone on relaxed muscle? Disappears, shortens, or disappear. Hope I'm making sense. That's what happens. If you don't believe me, go home and look at these things in your textbook or another textbook or in YouTube. There are nice videos. They show you what happens. Okay? So the cartoons, of course. And that's what. And then at the end, I forgot to say, at the end, all of these calciums cannot wander around in sarcoplasm. Sarcoplasm, same as cytoplasm. Remember that biology 1? Cytoplasm, well, we talked about it here too. So all of that calcium cannot wander around in the sarcoplasm. They have to go back to sarcoplasmic reticulum, and biology 1 again, when they go from high concentration to low concentration, they need ATP days, calcium pump. Or calcium pump on the sarcoplasmic reticulum, they have to put back calcium from here to here, and you need ATP for that. But 2 places you need ATP. 2 places you need ATP. When the head of the myosin, they do not have the head of the myosin detached, you need ATP, right? And also you need ATP putting back the calcium into sarcoplasmic reticulum. Okay? So, and of course, make sure it's nowhere in your notes in here what happens to I band, A band, Z line. What happens to Z lines? What happens to Z lines? Yeah, they come closer together when your muscle is contracted. The Z lines are closer together. Nothing happens to A band. H zone, I band also shrinks, where you have only actin between the 2 sarcomeres. Muscular movement, sliding filament atrophy, I talked about all of these. I just want to recap. Phosphate. What happens in your muscle cells, in your muscle, you already gave you enough creatine. Okay? So, but some athletes, as I said, they take creatine. You do have some creatine in here. So that's a reservoir. That's the term I use. A lot of textbooks use creatine as a reservoir. What happens in creatine, because you break down ATP to ADP, right? Biology 1. You break down ATP to ADP when you contract muscles. And of course, calcium is going back to sarcoplasmic reticulum. So you need more. That's not enough. What happens, of course, you have mitochondria, a lot of mitochondria in muscle cells that make you ATP. But again, there is another release for the shortage of ATP is creatine. Creatine attaches to phosphate, that is reserved, and then when you use all of the ATP and now you need more, the ADP attaches to the phosphate, I talked about at the beginning, attaches to the phosphate and then becomes creatine, and then ADP becomes ATP, and then ATP can be used to muscle contraction. I hope I'm making some sense. Is there any question? Does anybody have any question? I know I'm going through these a little bit fast, but it will help. It will help if you guys study these things a little bit beforehand. We will discuss them. We are discussing it in the lab, right? All of you, without exception. We're supposed to discuss it in the lab. How does a muscle contract? Remember, somebody could not pick up their muscle. What was the problem? One of the problem was nerve. The other problem was right here what I'm talking about. Okay, so when you come to the lab, we'll answer the questions that are coming up, the future notes. And that's what I'm hoping. We just glance at the PowerPoints, get them a little bit in your head. Okay, let's go. White muscle fibers. Homeostasis is the tolerable limits that your body can handle. For example, too much of heat your body cannot handle, too much of insulin your body cannot handle. Your body is under fluctuation constantly within tolerable limits, And that's the definition of homeostasis. Homeostasis, it means that and these are the systems that are responsible for homeostasis. All of your bodies, I don't like it when your textbook says these are the systems that are responsible. That's why this is me. This is me. You know, it means the rest of your, but your textbook says, these are the systems. Well, yeah, I guess it makes sense. These are the main systems contribute to your homeostasis. Okay, your body's homeostasis. Other systems contribute to your, even reproductive system contribute to homeostasis. But that's pretty much all of the systems. Okay. The first one, a few terminologies: extracellular fluid, further divided into extracellular fluid. It means fluid outside of cells. That's pretty much what extracellular fluid means. Blood plasma. So if I take your blood and put it in a test tube, what happens right here? If I take your blood and put it in a test tube, what happens right here are all of your red blood cells, and on top they call it formed elements. All of your red blood cells, everything is here, and right here it is your plasma. Okay, plasma. And right here, which is called formed elements, and I will talk about it in a minute. And the other thing, plasma, so is one type of extracellular fluid, and then interstitial fluid or intercellular fluid is the tissue fluid between the tissues formed from plasma. What happens if you have, here is a muscle cell, okay, here is another muscle cell, for example, or neuron, whatever you have. Okay, so the fluid right here between the tissue, between these cells, between the tissue, then is called interstitial fluid, which is a fluid outside of the cell. Outside of the cell is called extracellular. It's an extracellular fluid, first of all. Then that type of extracellular fluid is interstitial fluid or intercellular fluid. Okay, between the cells, between the tissues, and you have that in your body. Then you have another fluid inside of the cell. Remember that from biology 1? The fluid inside of the cell, I don't know if it's coming up or not, I might as well talk about it. It's called intracellular fluid. The fluid inside the plasma is called intracellular fluid. Like Highway 99 is an intrastate. It's just only in the state of California. The Highway 99 does not go to Nevada or does not go to Oregon. Does not go anywhere, just in California, Highway 99. But Highway 5 is interstate, right? It goes between California and Oregon and Washington, Highway 5, I-5. Just goes right here by your school, right? So it is an interstate. It's not intrastate. Intrastate. Freeway is inside of the state. But anyhow, just quick analogies I'm making. Then you have 5 different types of white blood cells, you guys. And then I'm hoping you would know platelets, thrombocytes, another erythrocytes for red blood cells, thrombocytes for platelets, and then you have 5 different leukocytes, which I'm hoping you would know. Sorry. These 3, I wish I had. These 3 are called granulocytes because they have inside of them, they have little granules. You see these granules, the granules in here is blue, black. The granules in here, sorry, the granules is here is red, yellow. And the granules in here, they do have granules, but they do not pick up acid or they do not pick up base when they stain them. On the microscope, without staining them, you would not know what they are, which one is which. But this one does not pick up acid or base, it's called neutrophil. If you look at the cytoplasm on the microscope, it's clear. But if you look at, I put them on display this morning, if you put these two on the microscope, one of them has dark granules, it's called basophil because they pick up base. Phil, it means love, like. And then this one is called acidophil or eosinophil. Eosin is the name of the stain they use. There are different types of stain. Your spinal cord that you have in the lab, it was stained with silver. We call it silver stain. There are different types of stain. Since the first time they looked at it on a microscope, they said, eosinophil. And later on, people like me came argue and said, no, it picks up acid. It must be called acidophil. So both names is correct for this one. Acidophil, eosinophil. So these are three granulocytes. It means they have granules. Amir, do we have white blood cells that do not have granules? Yes. There are two white blood cells, they do not have granules. Monocyte and lymphocyte. Lymphocyte is same size as red blood cell, but look, the cell is filled with nucleus. And you will know in a minute why. The cell is filled with nucleus. Okay? Then monocyte has a C-shaped nucleus, which picks up large materials, and then you will know the function of it here. And none of them, neither one of these two have granules. Neither one of them have granules. Let's go. Here we go. I wrote it for you guys. Interstitial fluid inside of the cell. Here we go. Intercellular fluid. Different types of white blood cells. Here it is. I would like you to know that. I don't expect you to know the numbers, but I will, based on looking it up there. If somebody asks you what is most abundant white blood cells in the body, which one would you say it is? Neutrophil. They are most abundant one. What is the least abundant one? Basophil. And then I would like you to know the functions. I'm not going to read it. But if you have any question,